So for the next session, we're going to go a bit deeper on the topic of data and metadata and ontologies, a bit more onto the details of actually managing them as a data steward. So first, about the difference between data and metadata. We've been briefly over this, but it's essentially a matter of scope. Okay, so we think about data as the object of research and metadata as data that's about the data. Okay, and an example I like to give is the clinical data. That for the clinical researcher it's the data, it's the thing that he works on, it's the object of his research. But for the molecular biologist, it's the thing that describes the samples that he is sequencing, for example. Okay, so it's mostly metadata. It's actually not very relevant to make a distinction. So in essence, what we want is that our data sets be self-contained and self-explanatory, and that means that whatever is not described at the level of the data and is needed to make the data set self-explanatory needs to be added as metadata. Okay, and so we often think about metadata as being external to data, but it can be part of the data file. Okay, or you could have data files with just metadata, for example, as we've seen in Bruno's data set. Okay, and actually in digital files, if you think about it, in the files in your computer, they come with metadata and it's embedded in the file itself. It's part of the digital signature of the file. Okay. And so in order to talk about what's needed to actually make data self-explanatory and self-contained, we need to think about the types of data. And so there are essentially three types of data, or 3.5. The first one, and it's actually the most common, it's unstructured data. Okay, so this is data that does not abide by any predefined data model. It does not have an organization in terms that you do not know what the elements of the data are and what are their characteristics, just looking at the data. And this means it's typically text-heavy data. Okay, an example is a research publication, so an article. That's unstructured data. Okay, of course we can think about a layer of structure. So articles have sections and they have subsections and they have an index and figures, etc. But that is not really structure or organization when it comes to actually being able to interpret the contents of the article. So the only way to interpret the contents of an article, short of using natural language processing, which is still not sufficiently reliable, is to actually read the whole article and interpret what's written there. Okay, and the same goes for written reports and books. And so the large majority of data that's currently available is data that is unstructured. Other examples are also images and videos. Okay, so images are unstructured data. So they have a machine structure in the sense that in your machine there is a default encoding, for example, a JPEG that encodes the color of each pixel. But that's not really an organization when it comes to interpreting what's in the image. Okay, so it's an organization in terms of enabling your screen to display the image, but not of telling you the content of the image. And both for images and videos, the only way to actually reliably interpret what's there is to view them. Of course, to get around this, we can annotate unstructured data. Okay, and we can do it, and it's commonly done for text, for images, and for videos, which is to add tags or other data elements that describe what's in the video, that describe what's in a scene of a video, that describe what's in an element of a scene in a video, or what's in a portion of an image, or what's in a portion of text. Okay, and this can be done typically manually, although there are machine learning algorithms that are able to do some automatic annotation of text and images and videos, and actually automatic image processing is getting better and more reliable than automatic text processing. And then we have structured data, okay? And structured data is data that is under an external data model generally, okay? So data model that's not contained in the data itself. And this data model organizes the elements of the data. So it standardizes what attributes the elements have, how they are connected to each other. Okay, and the most typical example are tabular databases, namely SQL databases. Okay, but we can also think about some typical data formats used in molecular biology as structured data. So FAST and FASTQ files which detail molecular sequences are structured in the sense that there is a predefined way in which sequences are encoded, and the file has a fixed structure. So it has a header for each portion of sequence that describes the sequence in a machine readable way, and then the data is the sequence itself. In the case of FAST, in the case of FASTQ, there's an additional quality score for each position. But this is it abides by a fixed data model that is the definition of the file format, and the information is structured and easy to read. The same for a sequence alignment file in the SAM format, or a genome file in GFF that describes the features in the genome, so the genes in the genome according to their positions, chromosomes, etc. So this is all standardized. There is an inherent data model, and the file is interpretable by anyone who is familiar with the data model. And then there is what is called semi-structured data, or self-describing data, which is data that abides by a data model, but the data model is described in the data itself. Okay, and common examples of these are XML data or JSON data. You see here on the right an example of JSON data. So there is a list of elements that is contained inside these curly brackets, and then you have key value pairs for each element, and sometimes you have lists of key value pairs inside some of the attributes, and you can have nested attributes. Okay, so you can have address, for example, has nested attributes inside it. So you can have hierarchy of things, and you can have lists of things, and the core is that your entities are described as key value pairs. So this is semi-structured in the sense that the description of the content is provided together with the content. Okay, and in principle, it's relatively easy to interpret, given that you understand the key value pairs. And you can complement this with a template, so a JSON schema, for example, that describes the fields that are allowed and the values that are expected in these fields, which in the sense would be similar to a database schema. So you can actually use XML and JSON in a fully structured way if you abide by a schema that describes what is possible to be detailed in your file. So in terms of reusability, in terms of being able to interpret what's in a file, and here we're talking about either data or metadata. So remember, the distinction is superfluous as far as we are concerned right now. So this structured or unstructured data could apply both at the data level or the metadata level. When we're talking about raw unstructured data, the reusability is obviously low. Okay, so the ability to interpret natural language texts, especially if they are large documents such as research papers, is low. So we have to expend significant effort into reading the text in order to interpret. When we have annotated and structured data, so if you have some description, some semantic descriptions of elements inside that unstructured data, then reusability increases. And it can increase substantially if the quality and the formality of the annotations is high, namely if we're doing annotations with controlled vocabulary such that we can combine annotations of different files because they follow the same standard. When we talk about structured data, the reusability is medium to high. The catch is that with structured data, you have to have the data model in order to interpret the data, and often you need domain knowledge as well, because the data model is not fully explicit. For example, if you see a database model, the database model tells you how each table relates to each other table, but it doesn't, and it tells you what columns you have in each table, but it doesn't really tell you anything about what the columns mean. So you need domain knowledge to understand that. Semi-structured data usually is high, often higher than structured data, because the data itself describes the data model. So you still need domain knowledge to interpret, but the data model is embedded in the structure of the data. And so that facilitates reusability to some extent. Okay, and when you talk about annotations, the annotations themselves are data. Okay, and so keep in mind that when I'm talking about unstructured data, it's not necessarily the whole file that's unstructured data. For example, in the data sets that you filled in on Dataverse, the comment field or the description field, which is natural language and accepts any length, that is unstructured data. Okay, so that does not come with any inherent structure, and you can write whatever you want there. So that is a portion of unstructured data inside what is essentially a structured description of your data set. And so annotations, even though they're generally small, so small pieces of description, they can be unstructured. Okay, you can add tags to a video in natural language. Okay, and that is low reusability. On the other hand, if you abide by a controlled vocabulary or an ontology, then you have semi-structured annotations, and then depending on the quality and how popular the vocabulary is, if it's used by across all databases for a given domain, for example, then it's easy to reuse and combine different data sets that are annotated in the same manner. Okay, of course, as we've seen so far, structured and semi-structured data are more reusable, but you cannot enforce only structured or semi-structured data. Okay, free text data fields or free text data sets are a necessary evil in research. And this is for several reasons. One is that you cannot model everything. Okay, you cannot have a data model that encompasses everything, and particularly not in research, because research by definition expands the boundaries of knowledge. So if you expect people to conform to an existing standard, they're not really carrying out research. If they're carrying out research, then they are doing things outside the boundaries and discovering new knowledge and potentially new techniques, exploring new ways of researching even. And so it becomes very difficult to create standards and models that encompass everything. And even if you could, semi-structured data, even if it's very rich and very detailed, never captures the full detail and intricacy of a natural language description. Okay, that's why we write research papers that are 16 or 20 pages long, and the data set has about 1 or 2 pages of metadata. So the solution is, especially at the metadata level, to compromise. Okay, you have to allow for some free text fields, even if you have structured or semi-structured data. So even in, that's why Dataverse has a description field or a comment field that allows users to input anything. And the other repository we saw, OpenNeuro, that Bruno showed, has that README field that allows you again to enter anything. Okay, so you need to allow a combination of structured data where you have controlled vocabularies, where you have fixed data types, and you have control over what is entered, and therefore are able to use those fields for finding data, for example, because you can integrate them in a search engine. You must be able to combine that with free text fields that allow the user to complement what is possible to describe in a structured manner with aspects that are not possible, either because they are particular to the data set in question and go beyond what's standardized, or because they are more detailed than what is standardized. Okay, and when we think about biomedical data, we have the problem also that there is a whole range of data subjects. So biomedical experiments go from the molecular level up to the organismal level, and they usually focus on processes, okay? And so in many cases, inherently, there is structured data, but also unstructured data related to these data objects, data subjects. Okay, when you talk about molecules, which could be drugs or metabolites, some of the information is naturally structured. Okay, so chemical structures, chemical symbols, molecular weights, etc. These are easy to provide as in a structured format. But when you talk about the roles of molecules in the organism, their interaction with genes, then this is much harder to provide in structured format because it requires a rich context and description. The same thing for macromolecules like genes and proteins. Okay, we have the ability to structure the sequence or structure information. Okay, but when it comes to describing the functions and the processes of the organism in which the genes are involved, then it's much more complex to describe information in a structured manner. For cells, again, the same thing. When we talk about the types of cells, we can structure that information. We can have an easy catalog of cell types. And then when we talk about the functions and interactions of the different cells, this is very deep information that requires natural language to fully capture. And the same thing across the levels. So for tissues and organs, we can structure easily the anatomy. There are controlled vocabularies for anatomy, for example, and compendiums of anatomy. But then when we talk about the functions of organs, then it becomes, and the interactions of organs, then it becomes very deep information that it's difficult to structure. Okay. And for organisms, we have taxonomic information. If we're talking about, for example, clinical data, we have the clinical background of the patient, so the age, date of birth, gender, etc. But then if you talk about clinical history, then that's again harder to provide as structured data. And when we talk about processes, then it's very hard to capture everything in a structured way. Okay, for example, just thinking about the gene expression experiment where we're comparing a tissue that has a drug and a tissue that doesn't have a drug, describing the whole process and describing the rationale underlying the experiment is not easy to do in a structured way. And that is actually where controlled vocabularies come in and ontologies come in. They are a means to try to structure some of the information that is generally unstructured. Okay, and we'll look at that in one of our case studies today, which is the case study of sequencing. Okay, so here we see a condensed history of sequencing in terms of the evolution of the volume of sequences available. Okay, keep in mind this is a logarithmic scale, so they have been growing at approximately a power law. So we started seeing public sequences when in GeneBank when it was founded in '82. So 5 years after Sanger first sequenced DNA, we started seeing automated sequencing in '87, and then first cases of whole genome sequencing in '95. Also, first DNA microarrays. A whole genome sequencing database was created inside GeneBank in 2002. The first cases of next generation sequencing machines appeared in 2005, and then in 2013 we started seeing third generation sequencing machines. So in terms of the data, genetic sequences themselves were represented as structured data from the very beginning because this is trivial to do. Okay. And in terms of finding sequences, of course, you can find sequences by their name, by the name of the gene, but this is not very useful because you'd need the name of the gene in the first place. The best way to find sequences is through sequence alignments. And early on, also sequence alignment algorithms were devised to enable finding sequence data. And BLAST is probably one of the most cited articles in history because of that. Okay, gene functions, however, are very different. Initially, they were only provided as unstructured data in the literature. So people would describe the function of a gene in the literature. But as the rate of sequencing increased, people started realizing, okay, we need to have some sort of controlled vocabulary describing gene functions. And so different databases initially adopted functional classification schemes to annotate their sequences. So in addition to the genome information, they would have annotations of those genes with tags corresponding to a functional classification. Of course, because these were different classifications for different databases, the reusability here was low. And in the late 90s, the several model organism databases realized this and pooled their efforts, got together and created the gene ontology, which essentially reconciles the different terminologies in use by the different genome databases at the time, and then grew to become something even more encompassing of gene function as a whole. Okay. And so that is precisely one of the main goals or one of the main roles of ontologies and control vocabularies is to try to structure to some extent, or at least enable structured annotations of data that is not structured. In this case, gene ontology is enabling the structuring of gene function. Okay. Although gene function is a multidimensional aspect, okay, and the gene ontology, for example, has 3 different dimensions. It considers the molecular function, the biological process, and then the cellular component where that function takes place. It's in no means fully extensive, so we can consider functions at different dimensions, okay, functions in terms of interactions with drugs, in terms of interactions with pathogens, etc., that are not necessarily encoded by the gene ontology. But still, it's better to have the gene ontology than to have no structured representation of gene function at all. Okay, of course, the function itself is not everything. How do we know how the functions are determined? Okay, or how do we know how the sequences are determined? In order to be able to reuse the data, we need not only to know what the data is, but how it was generated, whether we can trust it, whether we can use it to infer other things. Okay. So in terms of gene functions, currently the gene ontology annotations include an evidence code, so it's a 3-letter code that only describes in general terms the provenance of the annotation. So whether it was inferred electronically, whether it comes from a direct assay, etc. Okay, so this is in essence an annotation of the gene of the Go annotation that provides some insight into how the annotation was determined. Of course, this is insufficient for full reusability. You don't often have access to the source. In some cases, yes. Okay, when it's an electronic annotation, they add the source of the electronic annotation, so typically from Interpro or another automatic protein classification technology. But in many cases, you do not know fully. You cannot trace the source of the annotation. Okay, so you do not know how reliable it is or not. And for sequences, the information about the provenance is generally not included in the sequence files themselves. Okay, it's typically required when you deposit your sequences in sequencing databases such as GeneBank, but usually they allow free text, so it's not very structured information. And things get a lot more complex when we use sequencing, or in this case, microarrays to study processes. Okay. For example, if you're doing differential experiment using RNA-seq or microarrays, okay, what do we need for reusability in this case? Can anyone give me input? If we have a differential expression in let's say this example here, where you have cancer cells and normal cells, and you're doing a differential expression, what would we need to enable reusability of this data set? Can you give me examples of metadata?
Maybe the primers or conditions
are being used. The target genes, the level of expression, for example.
Yeah.
How the signal was measured, which signal. In this case, I think it's fluorescence, but depends if it's red fluorescence or green fluorescence.
Yeah, all relevant things. Also, who generated the data? What cancer the cells come from? How were the cells collected? How were they cultured? What was the process of isolating RNA? So in essence, to be able to reuse data from an experiment that focuses on a process, okay, so where we are comparing things and assessing the differences, requires a full description of the whole experiment, okay? Whereas, for example, a sequencing experiment where you're just determining the genome of an organism comparatively requires a lot less metadata. But still, if you want to be thorough, it still requires a substantial amount of metadata, even though that is not the norm. So in general, the amount of metadata demanded by sequencing databases is not as high as the level of metadata that should be provided. Okay, it's not just the equipment that was used for sequencing, but also the full detail of the sample that was sequenced, what was the DNA extraction process, etc. So specific.
May I
Yes.
just, so basically what you need is the methodology section that you would put in a paper, right?
Not only the methodology section, but also the author section and a bit more information than that, but mainly, yes, the whole methodology section. And preferably in a structured manner rather than as a full text description. Okay, and so this concrete case led to the arisal of the Miami standard, which stands for minimum information about the microarray experiment. And it has gone beyond microarrays, so it also encompasses RNA-seq since RNA-seq was created. So this was one of the first metadata standards, actually the first metadata standard in molecular biology. There were other standards in place for the medical community, but in terms of experimental biology, this was the first standard to be generated. And we talked about it previously. So in addition to the standard itself, they also devised a machine readable format in which to express the standard, because they acknowledged the need to provide data in a structured way to enable the interpretation of the data. Whereas in the case of sequencing data, the existing databases that contain very little metadata but allowed you to just blast your sequence against anything were sufficient for the demand of the time. The same was not true for differential expression database. And this led to the need for Miami. Okay, so on the topic of controlled vocabularies and ontologies, and again, like Miami was the first experimental biology standard, the gene ontology was the first experimental biology ontology, although there were other control terminologies and ontologies in use previously for the medical community. So in this sense, the medical community was a bit ahead in terms of standardization and formalization of the vocabulary than the experimental biology community. But we can think about a whole range of expressiveness from a taxonomy to a thesaurus to a conceptual model to incorporation of logical theory. And the expressiveness and the ability that we have to capture knowledge goes up as we encompass more semantic constructions inside our ontology documents. So the core difference between a controlled vocabulary and an ontology is that the controlled vocabulary essentially just concretizes the terminology used in a particular domain. So it restricts the terminology that is allowed in that domain to a subset of typically what would be used in natural language. Okay, and it can go a bit further than that by specifying synonyms and by specifying some relations between the concepts that are described. Whereas an ontology describes domain semantics. Okay, so it describes what entities there are and how they relate to each other and what attributes they have. In a sense, an ontology is an explicit data model of a particular domain. Okay, a self-describing data model. And as we've seen, we talked about this in the previous lab. So OWL is the standard for representing ontologies. Whereas if you want to represent thesauri, which are essentially controlled vocabularies that define synonyms and related terms, SCORS is the standard for representing. And we've been through a null exercise where, if you remember, we picked up an entity relationship model, and we went on protege and saw how to convert that into an OWL ontology that actually captured the semantics encoded in the entity relationship model. But that actually is much richer ontology than the typical biomedical ontology. So biomedical ontologies typically fall between a controlled vocabulary and an ontology. So they are very rich in the terminological component. So they define labels and synonyms, human readable descriptions, often cross references to other ontologies. And they have a semantic component, at least a class hierarchy, okay, of classes and subclasses, but it's usually relatively simple. So they are mostly a structure of classes, and they have very few annotation properties. Okay, they have a few annotation properties because of the terminological component. So they define things like label and synonym and description, which serve to describe the entities in the ontology, but they have very few object properties and no data properties, meaning they have few ways in which to relate classes to each other, except for the default class-subclass relation, and no ways to provide attributes. And another particularity is that in the biomedical domain, ontologies are generally used to annotate data, so they are seldom instanced. And what do we mean by this? So the key difference is that an annotation is when you assign an ontology term to describe a data entity or part of a data entity. Whereas in an instance, you have a formal representation of a data object as an individual in the ontology, okay, a member of a specific class in the ontology. So when you instance an ontology, you are essentially adding entities, individuals to the ontology. You are creating what is now commonly called a knowledge graph, okay, by describing entities in your domain as being members of your ontology. However, in the biomedical domain, this is seldom done. Okay, the majority of ontologies are only considered for data annotation. For example, the gene ontology is an ontology that does not have any instances. Okay, a gene is annotated to, for example, a Go biological process. It's not an instance of Go biological process. Okay, formally, we could have richer ontologies, okay, to express knowledge in a more self-contained manner. Okay, we could have in our ontology, in addition to the biological process ontology, we could have a classification of things like genes, and you could say that our gene is an instance of the class gene and it participates in a specific biological process. Okay, in practice, this is not how the biomedical community views and wants to use ontologies, and actually this is, I think, one of the hurdles between being able to use biomedical ontologies as vehicles to enable fair data more easily. Okay, because if you were to use biomedical ontologies for instancing data, okay, if you could express your data elements as instances of an ontology, then you could provide data itself in a semi-structured manner, fully described, that would not necessitate as much metadata as it normally does. Okay, and when it comes to annotations, the process, because biomedical ontologies typically use code-based identifiers and code-based URIs, so it's things like GO colon 000001. Okay, this means that to make your annotations human readable, in addition to provide the identifier of the concept you are using for annotation, you have to provide the label. Okay, so the actual function that corresponds to the gene ontology term. And that is indeed the norm, and this makes the process cumbersome. Okay, and this also means that if you were to encode your data in a machine readable format based on instances of the ontology, the data would not be human readable because it would rely on the alphanumeric identifiers. Okay, in the case of using ontologies for annotating metadata, the process is even weirder sometimes, because in many cases there is a textual field that is the basis for the annotation, and then the annotation identifier, and then the annotation label. But often the annotation label is exactly the same as the textual field. So this often generates redundancy. So this is one of the aspects that could be improved in terms of usage of biomedical ontologies to describe data and metadata. And the worst aspect, actually, of using biomedical ontologies is the tendency that the biomedical ontology community has to reuse ontology elements by copy-pasting. Okay, they will, be, in the interest of reusability, they will simply cut a piece of an ontology, the terms they want to use, and paste them into the ontology that they are developing. Of course, this is by definition an aberration, okay? This would mean that the concepts in an ontology have an independent existence on their own, which they don't. The whole notion of ontology is that it is a description of a domain under a particular point of view for a particular application. Okay, so the description of, let's say, human in an ontology that's for clinicians is very different than the description of human in an ontology that is taxonomical. Okay, and so it would not make any sense to reuse the definition of one ontology in the other. So this is really a problematic practice that has been become commonplace in the biomedical domain, and which I've struggled to combat without much success. And it leads to several problems. Okay, the first is that you find the same classes exactly with the same URI independently in various ontologies. And so what can happen is sometimes the original ontology changes the class, and then you have the same identifier being described in different manners in different documents, which is very problematic. And worse, when you want to actually annotate your data with an ontology and you use an ontology lookup service to find the closest term, you get multiple hits with the same exact URI. So how the hell do you choose which ontology to use if you have exactly the same term in 10 ontologies? How are you supposed to find which is the original ontology where that term is defined, and therefore which ontology you should use in your data set? So this is perhaps the most problematic point in terms of the current state of affairs in biomedical ontologies. Now on to metadata briefly. Okay, if you think about metadata in digital files, you can open any file in your computer and click on properties and then go to details to see the metadata that's embedded in your digital file. So this is by default structured. Okay, there is for each type of file there is a given list of metadata fields that is acceptable. Okay, and they are always key value pairs with expected data types. In some cases, you can add free value fields. Okay, you can edit these and provide large values if you want. And this metadata commonly covers authorship information, so who created the file and when, and a few file properties. So this is, I think, the most literal sense of the word metadata. So this is data about the data. For example, this is a Word document, so it tells me how many pages there are, what's the word count, what's the character count, what's the line count, etc. So this is the most strict sense of the word metadata.
Now, can I make a question, please?
Yes.
Just here in saying that is a structured data in this case. So if you have some fields that we have the name and we also have some free value fields, shouldn't be this be structured as we saw before, since it has the name, for example, for content type, pages, word count?
So what I mean with structure is that the thing as a whole is structured. Okay, so the metadata form is structured, but then you can have unstructured data entries inside a structured form. Okay, much like in an SQL database, you could have a free value field that allows users to enter anything, even though it's inside the database and it's a comment or description field. The content is unstructured in the sense that it does not abide by a particular data type. So any type of text is permissible. So what I mean with structured is that there are fixed keys and expected data types for most fields, but in some the expected data type is text and it allows anything. And so in terms of parsing that content, you would treat it as unstructured data.
Okay, thank you.
So now the question I was going to ask you is this sufficient for usability?
For me, no. I think the machines would like it very much. A user finds nothing useful about these, does not know what the file pertains to, what it has inside.
Exactly. So for usability, we need to know what the data is about, how is the data produced, okay, and how is the data produced encompasses characterizing the data subject. So if it's a sample, then how the sample was extracted, from what organism the sample was extracted, from what tissue the sample was extracted. If it's, for example, a sample of the plant, how the plant was grown, what weather conditions were in the field, if it's a field experiment, okay? So characterizing the subject can go up to the most minute details, especially when we're dealing with plants or animals that were grown in the field or in the lab for a long period. What the techniques were for producing the data from the subjects, how the data is organized, and of course, what we've seen, a license. What are the terms for data reuse? Okay, without which we cannot not actually reuse the data. So we can think about several layers of metadata. Okay, there is descriptive metadata, which is the one we use mostly for findability. Okay, this is information for identification and discovery. There's structural metadata, which is information about how the data is organized. Okay, there's administrative metadata, which is the technical source, data type, creation process, etc. Then there is reference, which is concerned mostly with the content and quality of statistical data. And then generic statistical information, as we saw, for example, the number of pages in a document, okay, distribution or outliers, etc. So in terms of enabling reusability, we are mostly concerned with the first three types, although to some extent also the reference. And of course, reusability requires structure, okay? To be able to interpret metadata, there must be agreement on what metadata fields should be present and how they should be filled, okay? And of course, they should be filled using controlled vocabularies wherever possible, okay? Free text fields should be avoided whenever there is a rich ontology that describes the concepts that are needed. Okay, an example, organism should not be fully free text. It should require you to fill in data, a controlled vocabulary term from the NCBI taxonomy, because it covers, in principle, all organisms and does not allow latitude. Okay, and obviously the role of metadata standard is to enable all this, to enable agreement on what metadata fields should be present and how they should be filled. Okay, and the problem with standards is that general purpose standards are structured and easy to fill, but they are not rich enough for usability. That was one of the things we saw today, is that in many cases the data sets we filled were not sufficiently richly described to enable full usability. On the other hand, no single standard can encompass all types of research experiments, okay? And in many cases, there are experiment types and research domains that don't have any metadata standard. Okay, of course, we can think about core metadata fields that are common to all experiments, but the problem is that specific fields then have different requirements when we go into the detail. Okay, and worse, even the cases that there are metadata standards, if you go through fair sharing, for example, you'll see that many standards are only available as a table of fields in a PDF document. Okay, they do not provide a data model. They do not have any semi-structured format for filling the standard. They just tell you these should be the fields and this is how you fill them, but they don't tell you any relation about the data objects. Okay, and also few standards are explicitly associated with control vocabulary, so they don't tell you which vocabularies you should use or are expected to use in each case. There are standards that have these things, so Miami is one of the best examples, but that's not the norm. That's one of the exceptions. Okay. There are some generic data models for research, namely the ISA data model, which stands for Investigation, Study, and Assay. but they are not rich or flexible enough to accommodate all experiment types or all research domains. So for example, and we will see in a few minutes, we deviated a bit from the ISO data model when we were modeling metadata standard for the plant phenotyping domain. And so that is the final case study I want to talk to you about briefly, which is plant phenotyping. So plant phenotyping is one of the most complex domains in terms of metadata descriptions, because, as I just said, we're taking samples from plants and we need to describe everything, including the environment where the plants grow, the provenance of the plants, if they came from seeds or if they are cloned from a parent plant, or if they result of the crossing of several plants. And the experiments themselves are very varied in terms of the range of things that are done. So there was already, when we started working on this, a minimum information standard called MyAPI, so minimum information about plant phenotyping experiments, but it was, as many are still in the current days, it was only available as a table of fields in PDF. On the other hand, the community also had BRAPI, which was a web service specification that enabled data exchange between databases. But this was developed independently from MyAPI, and so they were completely independent resources that even had differences in terminology between them. Okay, so we wanted to encourage the community to abide by a standard, but then we wanted databases to communicate using a different standard that was completely unrelated. And there were ontologies used to describe plant traits from the crop ontology that we wanted to encourage in MyAPI, but were not explicitly linked in MyAPI. So in my work in Elixir, and it was the work of, I think, about 5 years, we went through the extension and development of this standard into something that is machine readable and usable, and I think perhaps one of the best examples of a metadata standard currently in the biomedical domain. So we had to do a scope extension to accommodate woody plants, which were a concrete case of interest for Portugal. We had to improve the human readable definitions to make the standard self-explanatory. We had to add explicit ontology recommendations, and then we realized that we had to formalize the data model because it was not clear. So as a PDF of possible fields, what was the relation and what were the cardinalities between the things? I mean, how many plants can you have inside a study? How many studies can you have inside an investigation? How many publications can you associate with an investigation? These things are not all one-to-one, they are not flat, and so we need to have a data model that explicitly defines how they are related. And of course, then we implemented MyAPI in a number of existing platforms for metadata entry, in ISATab, in Dataverse, which some of you used, in FairDomSeq, which is a similar platform. And we went further than that, and we encoded MyAPI as an OWL ontology called PPEO to enable data and metadata to be published in RDF XML, so in a machine readable format as a knowledge graph. Okay, and when we did this encoding, we included an explicit mapping from MyAPI to BRAPI to enable the update of BRAPI. And we also are working on the encoding of MyAPI as JSON schema, similar to the OWL format, to encompass data publication in JSON. And on the side of BRAPI, we extended the BRAPI calls to fully cover MyAPI and made a direct mapping. So we used the mapping in PPO to make a direct mapping between BRAPI and MyAPI. And we also did the part of enabling BRAPI data to be expressed in JSON and in RDF, again, using the ontology version of MyAPI as data model. And last but not least, we mapped BRAPI to ISATab to enable importing and exporting of data in this format. And finally, we developed FAIRDARE, which is a which is a data discovery service that indexes data from all graph endpoints across the Elixir members and enables searches over them. So are we fair yet? Not really. So on one hand, incompleteness of ontologies hinders the enforcement. Okay, we cannot force users to use ontologies. We have to enable free text fields, but researchers often abuse them. So they tend to overuse free text fields and avoid using the controlled vocabulary fields. Ontology lookup, so facilitating the task for the user of finding ontology terms to describe their data set is not enabled in many of the implementations. For example, Dataverse does not have this facility. And in most implementations, there is also no smart filling. So if you have to fill in the same study multiple times or the same information multiple times in different studies or different aspects or different, for example, if you have different studies inside an investigation that have the same exact plant species, you have to fill the plant species n times by hand, whereas a smart interface would enable the reuse of this. So the vision for the future, and this is where I would like to take metadata standards, is to have, in essence, a service that supports both humans and computers with a graphical interface and with smart filling of data that is independent of the standard, and you can have a standard documented in OWL, for example, as an ontology, and you have an interpreter of the standard, and this feeds into whatever interface you have. So this is to some extent doable in Dataverse, but it requires manually encoding the standards in Dataverse. So it's not as easy as what I'm proposing here, having independent metadata standards as standalone documents that can be interpreted by any OWL parser. And the OWL parser has the advantage of enabling explicit encoding of ontology recommendations, but also data validation. Okay, so this is what I'm working towards, but it's still a few years away, I think. Okay, and that's it for me. We now have lunch break